Hello, everyone. Welcome back. Now we have Dr. Arun Zachariah from Kerala Forest and Wildlife Department. So the first time I met Arun in 2012, after my master's, I was working on frogs, and we wanted to do some phylogenetics work on frogs. So Arun's brother I was working with, he recommended, okay, maybe you should go and work with Arun. He has a lab in Wayanad, Kerala Forest Department. So I thought, okay, this is an opportunity. I can go there and study some frogs, things like that. But anyway, I was going on the way, I figured out Arun is not there in his office. So I had to stay in Calicut for a few days. Then after 2 days, Arun was coming from some tranquilization of some elephant or something, and he picked me up in the at 12 a.m. in the night, and we went to Wayanad straight to dart a tiger. So that's how the rate at which Arun was working at that point of time. But anyway, almost 2 years I spent with Arun in his lab. But this was an opportunity for me to actually look at, come into disease ecology and also kind of look at diseases in an environmental or ecological perspective. So throughout the time, or even before that, Arun was extensively work on wildlife diseases and epidemiology and disease ecology. So the time I was in the lab, Arun was working on these pathogens. He's going to talk about that today, I hope, something called elephant endotheliotropic herpes virus. These are the viruses which infect or maintained in elephant populations. So that was a really great experience, and I would give all thanks to Arun to take me along with him to the field of disease ecology, or show the field of disease ecology to me. And all thanks to Arun. And also, I'm lucky to have you here, Arun. Yeah.
Thanks. Thank you very much, Ansil. Thank you very much, Uma, Dr. Farah. And I'm meeting him after 20 years. Okay, so we were working in wildlife. So basically, I'm a butcher, glorified butcher. Okay, so I'm a vet. So I trained as a vet. I did my master's in wildlife biology, wildlife disease ecology. Then I did my PhD in wildlife biology. So I slowly transformed from a vet to a biologist, actually. It's my transformation. So to start with, sorry, I have a very bad cold. My voice is a bit raspy. This is a challenge, actually. So Dr. Guha extensively told the theoretical aspect of it. I think we can put some of our empirical data into it, so it's more interactive. Dr. Guha, no. So Dr. Guha and Dr. Farah and Uma can contribute. Everybody can contribute. I want to be more interactive. Okay, so this was 5 years back. This was the cover of the Time magazine, The Revenge of Microbe, when we have this all kind of MERS and corona outbreak and everything. They say, revenge of microbes. Microbes are coming back. In fact, interestingly, US Surgeon General in 1975 told, during the last 150 years, the Western world has virtually eliminated all infectious diseases. Okay, then we have to focus only. That was even CDC concept was changed, actually. So they was thinking the lifestyle disease is much more important, cancers and everything, diabetes, and the research and projects were focused to that. Nothing gone to emerging infectious diseases. Now the things has changed, especially after this post-corona situation. And 2000 even, Mahy and Brown predicted emerging zoonotic diseases are among the most important public health threats for the human. So this is the point of view, actually, the changing the world. Now much more focus coming into emerging infectious diseases. Okay, so for a biologist or an ecologist, disease is not something. We are looking disease in a different way. Okay, we are looking disease as a selection process. So most powerful selection process. Okay, natural selection process. So I can just simply saying this, this beautiful book by Anthony Sinclair, Serengeti Story. You know about Serengeti, okay, the massive, beautiful landscape, the most visited place of one of the national park in the world. You know who created that? A disease. Okay, Serengeti was fully occupied by Maasais, I mean the tribal people. It was not a wildlife sanctuary at all 100 years back, sorry, 150 years back. Then Europeans introduced cattle from India. So all of them were pastorals, you know, most of the Maasais are pastorals. So they want to improve the genetic quality of the cattle. So they introduced cattle from India to Africa. During the course, they brought one thing. They brought a virus. You know what is that? Rinderpest. It cleanly cleansed all the African cattle. So they have to move. So during the outbreak process, they pushed to the north and leaving this area blank. Okay, so slowly forests started to grow. Along with that, what happens? Tsetse flies settled in. So once the outbreak is over, when they're trying to coming back, you know, there is sleeping sickness. So they cannot occupy. Slowly it transforms into the beautiful Serengeti National Park. So a disease can transform a landscape. It's an ecology. Okay, that's the importance of the disease. So as an ecologist, I would say disease is more interesting. It's the selection process. I mean, I will simply say, there was 5 years back, there was a workshop in Delhi, a WHO workshop in Delhi. So they were arguing about addictive genes in humans. Okay, why humans have this kind of addictive genes and having more cancers than, have you seen the paper? Elephants doesn't have cancer, you know that, because the telomere is really long. Okay, but it's wrong. We have a couple of cases of cancers in elephants, especially TUSPL cases in elephants, but mostly in the wild, you cannot find much of cancers because it's undergoing natural selection process. But the human being, the problem, you know, we bypass the natural selection process. Okay, you know, the first important barrier, you know, there are 3 selection processes: pre-zygotic, zygotic, and post-zygotic. Okay, the first selection process in the human is the pre-zygotic, can be that, but there is no other evidence because the birth rate ratio is 1:1. I will come to that in the elephants. But when you look at the first one, is a pelvic. Okay, imagine, look back, 40 years back, what was the birth mortality in mother and babies? Now we bypass that. We have C-section, we have antibiotics, we changed everything. So for us, disease, we are looking in a different perspective. So it comes in that way, the One Health idea. Okay, so as I told, wildlife biology is a naive science. It's very recent. I mean, doctor can contribute more on that. So it's a naive science. It's developing. So they put the concept of One Health. You're all familiar about that. Okay, so coming into the emerging diseases, you know that 75% of recent emerging infectious diseases have been zoonoses, okay, from the animals, and 80% of them are from the wildlife. So you have this interface. You have the wildlife EID, human EID, and domestic EID. I'm working here in the wildlife emerging infectious diseases, and how they respond, how they interact with the human and domestic animals. So there are other factors: the global travel, human encroachment, translocations, encroachments, agriculture, technology. Everything is contributing to each dynamics. Okay, coming into this, these are the interfaces. We are talking about the interfaces because the disease has to be transmitted. There should be an interface. So these are the common interfaces, they say. Live animal market, as Dr. Said. Then wildlife hunting, the domestic animals, and intensive wildlife farming. It's again coming up. So these are the major interfaces you can find. Okay, so finally, we come to this wildlife disease ecology. We look into disease in a different perspective. It's an ecological aspect. Okay, so we are looking into 3 factors: the agent, which is a microbe, and the host, and the environment. So this is a triad. This is called ecological triad. So this triad is forming the disease ecology. Okay, so environment has a role, host has a role, agent has a role. Okay, agent can undergo changes, mutations, interfaces, and environment. Climatic changes can enhance this, and host, genetic structure of the host can change this. I will come to that with some examples we have. So what we did, the disease in wildlife, so I told you, can be just classified based upon etiology. We can look into as microparasites and macroparasites. Microparasites are just, I want to be more interactive. Please, if you have any, this thing, just raise your hand and just shout to me. Microparasites are, virus, bacteria, fungi, those small pathogens. And macroparasites are, the flukes, insects and those kind of things. But in epidemiologically, we can just put it like this. Okay, so it's the kind of classification we did. So we say inherent diseases. Okay, these are diseases evolved along with the species. It's called coevolution process. Okay, then there are some diseases. I mean, Dr. Guha told about that. I can give you some example about affecting multiple taxa. The dynamics is entirely different. Okay, I will come to that again. And we have finally the emerging diseases. Okay, we look into that each cases. For example, this is the stomach of an elephant. Okay, as I told, I'm a glorified butcher. We did more than 580 postmortem in elephants so far in the past 20 years of my career. So, see, this is stomach of an elephant. You can find the boats here, right? Can you see? Maggots. Okay, these are maggots of a fly called Coboldia elephantis. Okay, this maggot, this fly lays eggs on the commish of the mouth, and later it's swallowed. It hatches in the stomach. It becomes maggots. It becomes pupae and become a fly and flies through the mouth. This is the only place where this fly can breed. Okay, this is ultimate coevolution. You cannot find these maggots in other animals, not in gour or in sambar, nothing. Only in elephant. Okay, this is the ultimate coevolution. It's, yeah. Yes. Coboldia, yeah. They're similar species, but elephant is in Asian elephants, and the other one is Coboldia. I just forgot the species name. So again, the genus is same, Coboldia is same. Yeah. Please.
Is that insect completes its life cycle within the living?
No. It just only breed. After that, after pupates, it breaks the pupae and it flies through the mouth.
Up to pupae, it stays within the host.
Yeah.
Living host.
Yeah, a living host. A living host. So, and interestingly, it doesn't cause any disease at all. If you cut upon any elephant, you can find this in the stomach. It will not cause any harm to the host. Yeah. That's I'm coming into. Okay. Can't say it's a commensal. Of course, it's a parasite. I think Dr. Guha can contribute much on this. It's a parasite. Of course, it's a parasite. It has its own negative impact on the host, so definitely it's a parasite, but can't be a commensal. We can't say it's as a commensal, but does not cause any disease at all. Okay, so, yeah. Sorry?
Gloves.
It's an old photo. At the time, we were not using gloves at all. Okay. After we found the TB and everything, we are having whole suit, and you can do that in the forest most of the time. It is really hard to do it. Anyway, I'll come to that. Okay. The next case is it's called Grammocephalus species. Grammocephalus or Grammocephalus we can call. So this is a parasite affecting the liver. Okay, it's only seen in elephants, and it causing the maximum mortality in elephants. Okay, especially calves. 60% of elephant calves are dies simply because of this Coboldia elephantis. Grammocephalus viridatus. So it's again very species specific. At the same time, it kills. Okay, Coboldia doesn't kill, but it kills. Okay, here we comes another concept we call density dependent selection process. Okay, it actually controls the population size. Okay, when you look into elephants. How much time I have?
Not 2 hours. Well, now you have 1.5 hours.
Okay. Okay, so look into the elephants. So we took natality. So okay, there are two parameters we can look into survivability. Which are they? Natality and mortality. Okay, so we looked into natality. We found in Mudumale they had 135 birth records in elephants. And we have the US data and UK zoo data also. We found that the birth ratio is 1:1. So there is no pre-zygotic or zygotic selection process, like in crocodiles, when the eggs, the climate change, the temperature change, the sex ratio varies, okay? But 1:1. That means there is no pre-zygotic selection or zygotic selection. But look into the wild, you see, it's highly skewed. 1:40 in some places, and 1:100 in some places. She recorded in Periyar. So 1:100, highly skewed. Why it's happening? Why it's happening? Here we say, this is the one. He controls, disease control. This parasite controls the selection process. Okay, so we are coming into that into detail. Look into the another one. In the case of tigers, we did a lot of postmortems in tigers. This is one of the main reasons for their death. This is called, we call it time bomb in the tiger. This is called Galungus perniciosus. Oh no, sorry, I don't have the detailed picture of it. So these are just nodular parasites. I had the slides, but unfortunately I didn't put it. Okay, these are the parasites affecting the gut intestine. And when you cut open any tiger or leopard, you can find this. Okay, nodules. Okay, when the immunity goes down, if the animal is aged, they started rupture coming out, breaking the leakage of, causing the leakage of intestine, and dies due to peritonitis. Okay, so this is, the tiger is upper predator, only except human disease is the one actually it controls the population. So this is one of the major causes mortality in tiger. That's why we say it's an evolutionary process, most of the disease conditions. This Dr. Guha told the mortality due to microparasites, multiple attacks. For example, in the case of anthrax, this is a typical case of anthrax. We used to get anthrax, not in outbreak proportion, but sporadic in every year, we get 2, 3, 4 cases of anthrax, which not like an outbreak. Africa, they got an outbreak, right? They got an outbreak in Africa, anthrax. Dr. Farah, yeah? Oh, 10 years back, they got a huge outbreak, but we are not getting it in an outbreak proportion, but in a very sporadic proportions, we are getting the anthrax. So it's affecting multiple taxa. It's affecting the cattle, it can affect goat, it can affect the deer, and it can affect the elephants also. The spores which are living in the soil can, transmit the disease. So the dynamics are entirely different. So finally, we come into wildlife disease ecology. We comes to the coevolution and niche. Okay, this is a classical example of niche. It's a cassinophorus disease. Okay, so we reported first in 2014. We had a huge outbreak of cassinophorus disease in Kerala, killed almost 350 monkeys. Around 200 human were infected. Okay, 15 died because of this thing, but now it's endemic. Previously, there was no record of KFD in Wayanad. I will talk about it because what happened? We have a KFD in, this is the history of KFD. In 1955, the first case reported in Shimoga, in Casanudi village in Shimoga. And because there was a huge harvesting of trees and there was a close contact, it's transmitted through the ticks, okay, tick. So they got the first incidence in 1955, then there was huge mortality and become endemic into this thing. Our story was different. Now, this is actually the Casanur, and here we have our outbreak here. Okay, so when we did, so these are the status of. Then we have the Chamarajanagar district. There was another outbreak. Then we have the Wayanad outbreak. The first, we got the monkeys coming out, dying, and we have the massive hemorrhagic lesions because we do the postmortems and massive hemorrhagic lesions. At the beginning, we were surprising why it's so, and the story behind that. And actually, we did a sequencing. Okay, sorry. We did a sequencing of the virus. Okay, so Ansil was at the time, right? No, you were not, right? So this is the cluster. This is our cluster. Okay, so you can see that clean divergence. 8% nucleotide level difference between the original KFD isolate. Okay, so why it's so? Why the difference? You can say that it undergone mutation. Chance is less. Okay. What we did in 2013, there was a court order. There are a lot of small towns in Wayanad, and there are a lot of monkeys there. Okay, resident monkeys, and court said, catch these monkeys. It's a menace, and release them back into the wild. So Forest Department, what we did, we captured around 3,000 monkeys. We released them back into the wild. Okay, within a month, we have the outbreak. Yeah. Yeah. Exactly. Exactly. So that's why we abandoned the mutation theory actually. Okay. Yeah.
In case people don't use them, I just repeat the question.
Sure.
The recording people
Sure.
can listen later.
So I want to repeat the question.
No. Say like she said something, right?
Okay.
That won't show up in the recording. You can just repeat.
Oh yeah. Okay. So that divergence is quite interesting. That's why we thought in the beginning it was a mutation. That's why we got this. And either by the tick expansion, I will come to that because, this could be the possibility. The expansion of range of ticks or vector may be due to climate changes. So it just spreads across. Anyway, it's connected. The landscape is connected. So expanding range of reservoir hosts, that's the question. Is it a mutation of the virus, a new virus strain or exists in the landscape? There we pointing out. We did it. Sorry. Yeah.
So after you just captured that all monkeys, did you vaccinated that monkeys and after you left?
No. They have the box traps in the city, in the towns. So it becomes a big menace. And court ordered just to release it back. Useless thinking. Okay, so we captured 3,000 monkeys and we released deep into the forest.
Okay.
All of them came back. Okay, all of them came back. Monkeys are amplifying hosts for KFT. What it did, they picked up the infection. They came to the human settlement. Then we have the massive death of monkeys and the human infections. 200 infections and 15 death, human death.
Is there any vaccination drive for these monkeys?
Yes. We have vaccination. There is vaccines developed and we are now extensively vaccinating in the interface area. Human.
To the humans or the monkeys?
No. Humans. You cannot do vaccination in wild. Okay. So it's.
They are actually from Coimbatore University, and they are working on KFT, sir, and his two students.
I know. She also told that. Yeah, wonderful. So it's quite interesting, actually. So that's why we said, here is the point. So mutation rate is actually extensively different, 8% of nucleotide differences, and expanding the range of reservoir host dynamics, that's exactly we have to answer. We don't know the reservoir for the KFT, okay, which is really hiding. And interestingly, I will tell another story also. 5 years back, there was a WHO meeting in Mumbai and talking about the rabies. Okay, so there were human doctors, there are veterinarians and wildlife vets like me, and they're arguing. Human says it's a human disease. Okay, veterinarian says, no, it's a veterinary disease. As a wildlife vet, I say, it's a wildlife disease. Okay, still it's confusing. Where is the reservoir of bat? Everywhere in the world, they discover the reservoirs. Okay, in the bats and also in raccoons in US. Okay, we don't still know. So spillover is happening. Okay, it's spillovering from the host to maybe a fox or maybe a jackal and transmitting to dogs and coming into human. Okay, human is the end host. Okay, so that kind of mechanism. So we really thought this thing, there are niches there for KFT in whole across Western Ghats. Okay, and the amplifying, they're coming out and coming into interface because we did that. So we've created the interface by releasing these monkeys back into the niche, and they got it, they amplified, they came back to the human. Make sense? Okay, so that's why we call disease niche. As coevolution goes on, disease comes to the host and become adapted to the host and form a small niche. Could be ecological, could be a species-wise, very fine defined niche. Any change in the niche actually increases the interfaces. Okay, either due to vectors or due to climate change or whatever things. Okay, these are actually when we go to in disease ecology in wildlife, we say they are co-evolved with the species, and we have to look into interfaces, what really propagating them coming into spillovers. Okay, so another example, in 2016 actually, we have huge mortality of elephants. Okay, adult elephants are dying. You can see, usually, when in the wild, when you do necropsy, when the elephants are really chronic, sick, or old, where you can find the carcass? Anybody can guess? Near to the water body. Okay, because they are weak, the primary thing is to get water. Most of the death you can find near the water body. Okay, look at them, all of them, you can see them just dying like like an apple. Okay, just they're falling. Okay, nothing needs peracute death. Okay, you can differentiate a carcass from seeing whether it is shot on the brain or the disease. You can find the position. If it is shot on the brain, you can see it's in a sternal position. Okay, if it's a chronic disease, you can find the lateral position. You can find a lot of struggling and everything. Anyway, so we had this outbreak, like massive outbreak, more than 120 elephants in our landscape. Okay, so you know the problem with the I will come into that. Okay, actually, so how we can investigate a wildlife disease in real wild. So what we did, so first we thought it's anthrax. Typically it's look like anthrax. See, the carcasses, you know, it's within hours, it blots like anything. Okay, exactly like anthrax. So we avoided a couple of cases in the beginning. It was a summer. So, but what we did, we tried the spot PCR for anthrax and it was negative. Okay, then we decided, let's cut open the carcass. We cut open the carcass. Oh, sorry, I missed those slides. Okay, we cut open the carcass. We found it just like, typically like anthrax. It's something anaerobic infection is going on. Okay, then we try to do all kind of classical mechanisms like staining and everything. You know the problem with anaerobic organisms, when the anaerobes get in, all the anaerobes will be supported. You cannot differentiate from a single staining slide. Okay, so we try to do everything. Then finally we thought we can go for metagenome. Okay, this is maybe the first example to use metagenome in wildlife disease investigation. So we did the Illumina metagenome, V3, V4 region. You know that, right? Okay, bacterial region, highly variable regions, V3, V4 region. We amplified, and we got, whole signature for Clostridium novae. Okay, Clostridium is not at all reported in wildlife, I would say, except in bighorn sheep in Canada, and only in some domestic animals, in pigs and horses and sheep mainly. Okay, so it was a Clostridium, and then later we did. We develop a primer for the flagellin gene. We find that it's a Clostridium novae type B. Okay, so based upon 16S RNA annotation, we found that the phylum at the species level, we found that through the samples, and we got 298 samples and 232 was Clostridium novae positive. We got our archived samples also. Okay, so it's the first report of Clostridium novae. So we are surprised how they are getting this infection. Okay, it's just like the last year also we had a huge mortality. Okay, so during that time there was a massive mortality of elephants in Botswana. Okay, so they said it is due to phytoplankton toxicity. Okay, but here, so when we presented this thing in 2 years back in Thailand, so they were quite interested, and they're looking into, they're getting Clostridium as well in most of the cases. So when we did that, we just want to know how this thing evolved. And so we found that we have water bodies. Okay, so we have two types of water bodies, man-made water bodies and natural rivulets are there, or rivers are there. So we found all of our man-made water bodies are contaminated with Clostridium novae. Actually, it's forming a biofilm. Okay, so this is actually induced by us, right? So we stopped doing man-made water bodies nowadays at all in our landscape. We totally avoided that because this is a kind of poison, you know, it accumulates and it comes up. Actually, it got a clear relevance with, you know, climate change also. Clostridium novae proliferation, you know, the waterborne diseases are going to be the biggest threat for, you know, the emerging infectious conditions actually. Okay, there are lots of diseases coming up through the waterborne conditions. And so it's the first environmental contamination of pathogen we are recording as an emergence. Okay, now it's very clear. And mortality in other populations were identified with similar lesions. Like we got samples from Karnataka and Tamil Nadu also, and we are getting the outbreaks as well. Okay, so is the climate change, can we connect with the climate change? It's very difficult. Basically, it's very difficult to prove it. But with our, you know, pattern of epidemiology, because we are getting this infection during summer season, okay, not in the rainy season. Okay, so it's very clearly there is something happening, and can be connected with the climate change. That's what we are doing because we need more data. Okay, so I told you about the pathogen, okay, environment, and coming to host. Okay, this is called a disease called endotheliotrophic elephant herpes virus. Ansil worked extensively on this. The disease, I would say, first reported in US. The first case in 1972 in Switzerland. Switzerland. Ostent, one scientist, actually, he did a necropsy on a circus elephant calf, and he found herpes inclusion bodies in the histopathological slides. So he said there is something like herpes virus in elephants. Then the second case was actually recorded by one of my colleagues. So she worked in US in National Zoo. So they had a baby elephant calf, Kumari, the first artificial inseminated elephant calf. Okay, she died suddenly. So Laura did a necropsy. She found massive lesions. I will show you the lesions. And they find the elephants are dying because of a herpes virus, new herpes virus. So these are the lesions, peracute death. Okay, peracute death, these are in the captive cases. You can find your elephant calf happy, and within 24 hours, you can find she's moody, she's not moving, she was sluggish, and within 24 hours, it's dead. Okay, within 24 hours, very peracute, and the lesions are massive. Okay, so they found when they did the retrospective sampling, the sample analysis in US zoos and UK zoos, they found that 80% of the elephant calves are dying because of herpes virus. Okay, then she recorded. Meanwhile, they find almost a similar virus, the herpes virus, okay, EHV-like virus in African elephants also. Okay, African elephants, no mortality is happening. So they have some nodules on the trunk, okay? And these papillomas contain this virus, herpes virus. So the hypothesis is that this virus might have been jumped from African elephant to Asian elephants, because they are keeping African elephants and Asian elephants in clear vicinity. Okay, then we reported actually the first cases in the wild, in the wild. Okay, so this was a disease considered in the captivity. Right? Okay, then we got a case in the wild. All right, so these are our index cases. Integrity, right, Ansil? Right. These are our index cases, and now we have large number of samples. Then we sequence the samples. So these are the clinical signs. Peracute nonsense, you can find fever, anorexia, dyspepsia, means not eating. And you can find the facial edema in all the cases. Just face will be puffed off. And blue tongues, okay. And the pathological, extensive hemorrhagic lesions, petechiae of the pericardium, pericardial effusion, petechiae of the peritoneum, hepatomegaly, massive lesion. I can show you. This is a facial edema. So puffy face. Okay, you can feel that. And the tongue would be is bluish in color. It's called cyanosis. Okay, and the massive lesion. This is heart. Okay, see the epicardium outside of the heart. You can massive hemorrhagic lesions. Okay, I would compare with Ebola actually. So even it's a DNA virus. And the liver would be just like puffed off balloon with subcapsular hemorrhages and massive lesions. And even in the brain, you can find hemorrhages. So this is a typical inclusion body of the herpes virus, okay? Amphibilic inclusion bodies. Then we recorded whole across Western Ghats the first time. Then we published the paper. Actually, it was a shock for US, okay? Interestingly, there was a huge campaign against zoo elephants because of this herpes virus, because they say they're killing elephant calves because they are mixing elephants together. Then we came up with this herpes virus in the wild. We sequenced, and the whole team came from US to us. Actually, we saved the zoo's elephants from US because we said, no, this is not a disease transmitted jump from African elephant to Asian elephants. We did a lot of sequencing, including the whole genome sequencing. And we found, this is actually what happened. We proved how it is transmitting. Actually, this elephant calf was our index case in one of the captivity. And his name was Niranjan. We rescued this calf. And after 4 days, the next calf in his vicinity got infected. So we sequenced the virus, we found 100% similarity. So we proved that this is transmission through close contact. Okay, later, we found that adult elephants are shedding this virus, herpes virus. Okay, so literally, then in the wild also, this was our first case in the wild. No, fourth case in the wild. Actually, we tracked this herd for 6 months on foot, our team. Okay, we found the same mortality in the same herd, and we compare the virus, exactly the same. Okay, so it's changed everything, all the perspective of it. So when we sequence, we find that, sorry. Okay, so when we sequence, we find the same isolates in US. Okay, EHV1A. There are so many EHV1A. We look for different variable regions and also conserved regions. Looking at variable regions, we find the same isolate from the US also, Europe also. Okay, so finally we proved that it's not a emerging disease. It's a disease co-evolved. When we make the tree, we found that it's almost parallel to elephant evolution. Okay, so 10 million years back, elephant moved from Africa to Asia. You can find the same pattern. Okay, so it's actually the co-evolution. Okay, so literally what we're saying, every elephant born is under the shower of herpes virus because it's shedding. Adults are shedding. There is no clinical signs in the adults. Adults are shedding. Some adults are shedding. Okay, some of our camp elephants are really shedding, so we isolate them. We never let even our mahouts to come in contact with them. Okay, so every elephant calf in the wild born is under the shower of herpes virus. Why some of them are dying? This is a question. Not all of them are dying. Some of them are dying. That is a real challenging question. So, and again, why we recorded this thing? The first case actually, I told you in 1995, first case in National Zoo in US. Then our case actually, Dr. Krishnamurthy sir, he reported one case in 1997. But unfortunately, there is no PCR system at the time. So we cannot send, the German team is really keen to have it, but we don't have the permits to send a sample, so it was undiagnosed. Then we come up and we reported the cases in 2007 onwards. Okay, so then we have an extensive lab for this alone, for herpes virus itself. So we got US Fish and Wildlife funding, and we developed this lab. So the question is, why it is not being recorded? You know, the elephant mahout tree and the management is extensive. There are lots of work did. Okay, then after that, we find the same thing in Indonesia also. We had a project there. We found the Indonesian calves are dying. Because I think Sumatran elephants are going to die because of elephant herpes virus alone. Okay, then we have in the Myanmar. Now we have got in Sri Lanka. Everywhere we are recording right now. Okay, so what we think, why it's not being recorded? See, such kind of massive lesions. Okay, quick death. Okay, can happen because of, you can say it should be a poisoning or something like that. So quick death and massive lesions. There are extensive lesions and the hemorrhagic lesions also. How they missed it? How they missed it? Is it not possible? Because we have a huge history, you know, records. I dig up records in British Library, and I couldn't find any, because there are huge data of elephant birds in Northeast area and everything. They never been reported. But somehow it's coming up. It is increasing in prevalence. Okay, so we think so many populations going to extinct because of herpes virus alone. So what's really happening? To look into that, we are looking into host genome. Okay, we find all these cases have a low genetic diversity. Heterozygosity level is very low. We're working on it actually. It's a preliminary thing. We submit a paper for publication, and we found all of them, okay, the whole DNDS data is really homozygotic. Okay, so this may be a reason. I can't say it right now, okay, unless and until we have more specific areas to look into, and because we are looking to immunological genes, basically. So this could be one of the reasons. Okay, all the populations undergoing because we killed, you know Hamilton-Zook hypothesis, right? You know that? Sexual selection? Okay, why peacock has a big tail? Because it's a disadvantage for him. Okay, any tiger can step on it and kill it. So negative advantage. So sexual selection is something interesting, quite interesting in wildlife. It's one of the major forces for selection. I think Uma can explain much more on that. So it's a handicap. It's called handicap hypothesis, put by Hamilton and Zook. And elephants having this is called secondary sexual characters, okay, because female is investing. Okay, look in the case of elephants, maximum birth would be around 6. And quite interesting, I would say 60% of females are not breeding at all in their lifetime in elephants. When we do the postmortem, we look into the scars, birth scars. 60% of females are not breeding at all. What are they doing? They're supporting the family. Maybe, we don't know. Okay, so look into that. Only 6-7 birth in the lifetime. Okay, effective population size would be, I would say, how much, Uma, I would say, in our, yeah. Nobody did it, we are doing it.
Yeah, it's difficult to say, but several thousands probably.
In our Eastern Ghat population, you say?
Western Ghats,
Western Ghat population, I would say.
several thousands. But I won't wager how many.
There are 9,000 elephants in our landscape. Yeah.
So you said because of low genetic diversity, some of the populations might be more prone to the disease aspect,
that's
right?
a hypothesis.
It's a hypothesis. I mean, I've seen some of the talks. We had a wildlife ecology meeting like just a few weeks ago, and they showed a lot of data to do with how the elephant population, this is in the southern India, they are also very affected by the drought and other cycles, which affects their nutritional aspects, like how some of the females, they don't breed because they have such a long gestation period also, like 18 months or something. And when I looked at that data set, this is like done for years and years, they have followed some of the herds. I felt that where is the disease aspect missing? I just feel those two things needs to be combined in a way to understand some of these things better. That's my one thing. And B, for a species like this where we can collect the dung, I think the monitoring for this herpes thing can be done through the dung also, right? Like which herds are, as you said, adults do shed most of the time, or maybe it increases some time of year, or I don't know. Some of these aspects needs to be tied in with the larger picture, I guess.
I'm sorry, I didn't explain that actually. We got from the trunk wash.
Okay.
Okay, through the trunk, the virus is escalating.
Through the
trunk?
Yeah, but I mean, you are the...
Not through the dung.
Okay.
The adult one, which are carriers, they shed the virus through the trunk.
I see.
Okay, so they communicate with the trunk.
I see.
When the first elephants comes, what they will do, they will touch it and put it into the vomeronasal organ. Okay, so the transmission is through the trunk.
No, so that's a mode of transmission. I meant in order to follow a herd, there must be some traces in the dung as well, right? Which can be monitored, looking into what herds are more prone and whatever.
Virus is very fragile.
Sorry?
Virus is very fragile.
Fragile, okay.
When you come to air contact, it goes.
Oh, I see, okay.
Okay, so that's why we're getting all these cases from the postmortems. Yeah.
Sorry, I just want to add one thing. Sorry, I was zoned out. In terms of elephants, you want some water?
No, it's okay.
In terms of elephants, it's not yet accepted, but we have this paper in revision, which is basically showing a north to south colonization and dilution of genetic variation as you go from north to south. Yeah. So basically, elephants probably came into India from that, northeast area.
Persia.
Yeah. So basically, as they come south, you see less and less genetic variation.
That's what's happening in Sri Lanka.
Yeah. So what happens then is, in fact, in the Western Ghats, you actually have 3 populations: north of Palghat Gap, south of Palghat Gap, and south of Shenkota Gap. There are 3. We suggest different management units, and there's different variation, lower and lower variation. Yeah. That also corresponds to lower and lower population size.
Exactly. Exactly. Yeah.
But that's just broad genome-wide variation. Yeah. I'm not commenting on disease susceptibility or maybe it correlates. It's something which needs to be investigated.
Yeah. Exactly, she told. That's kind of genetic diversity dilution happen as, elephants, just like humans, they came through that and they occupied slowly. Yeah.
Is that, as you said, in USA and UK zoo, elephant died because of the same virus and also in wildlife. Is that mortality rate is same in zoo as well as in wildlife?
say, I'm coming into that actually.
Frequency.
Wild is very difficult, you know. I will come into that. What are the challenges when we do the wildlife disease ecology?
Is that correlation of spreading the disease?
We did this training. Okay, genotyping. We found the same genotype in US. That's why when we have the same genotype, we were surprising how this is come into it. Actually, there is no African elephants in our vicinity except in Mysore Zoo. Okay, there is only chance for, but it's in total captivity. There is no chance of spillover into wild. We are getting those things all across this Western Ghats landscape. So this is the same question. So why we answered that, you know, in Europe and US, they pick up the wild elephants from range countries. Each range has their own strains. Okay, so they pick up them. By doing that, they have a pool of vat of virus strains. It's simple, because they brought the virus from different. Indonesian strain is actually exactly different from Thailand, and also from Malaysia, and also from Sarawak, and also from Burma. So which are different.
Excuse me, in general I'm asking.
Sir, did I answer that or?
Yeah, it's right. But is there any solution strategy to increase the diversity by pooling animals from different places? So in a longer perception, just to save the wild.
We are coming into that actually. We don't know.
So because somehow you have to conserve the wildlife.
This is actually,
so otherwise it will go. Simply it will go just by within a short period of time. Instead, can we shift animals from different location of wild? Thereby we can increase the diversity as far as genome is concerned.
That is a probability, because still it's only a hypothesis actually we put forward. Okay, that is a possible hypothesis. Actually, we are looking into immunological genes variability, and we find that there are lots of immunological homologous genes, and diversity is very less in herpes viruses. That's why we are putting forward, could be the reasons,
So what you're talking about is a strategy which people are using now called genetic rescue, where you might want to enhance either total genetic variation or specific genetic variants. For example, if you know a specific allele is protective against a disease and you don't want the disease to wipe out the species, that's a conscious decision you're making though, then you could probably think of enhancing the frequency of that allele by bringing in individuals.
That's the concept of metapopulation, Uma putting forward, and making the connectivity in between populations. So that's the crux, and she's working on it.
So just to, finish that. For example, there was an outbreak of SIV, simian immunodeficiency virus, in chimpanzees. And researchers had samples prior to the outbreak and after the outbreak. And they were able to actually identify specific loci which seemed to be selected. That is, they would infer that individuals with those loci were able to survive SIV. That is the kind of information you need to make those decisions. Otherwise, you just say generally will increase variation, but you have to have a lot of detailed information.
Yeah, but basically when you look into DNDS data, heterozygosity, we found that these clusters, herpes clusters, have a very low genetic diversity. Synonymous to non-synonymous substitution rates are very low. Okay, now we are going for specific genes, immunological genes. Yes.
Maybe,
please, sir.
You may explain, I expect the same. Just, it is. Cheetah, for example. So the cheetah's population is vanished because of one incidence of virus. I hope you know that. In zoos, just they realized the reason is just the narrow diversity, the lack of diversity with respect to the loci of MHC. MHC. It is recently discovered.
We are applying the same thing.
I'm expecting the same.
MHC1 we are trying.
So I thought, so the same thing we can explore in future.
I will come to that, because what sir told is very relevant. So that's why I'm saying, look into the risk aspect of elephants. You know, a female can give birth around 6 maximum. Okay, and again I told, all the females are not breeding at all. Okay, they are helping the herd. Okay, so with our data. So how they maintain the genetic diversity? Like this large, they got a huge lifetime. how they're maintaining, elephants are maintaining the genetic diversity. Can you imagine? Ants can do that. Arthropods can do that. How this larger mammal have very limited chance of having so many progenies, okay? How they are maintaining the genetic diversity. That's the risk of extinction of megafauna. Okay, here we are coming into that. Okay, it's look into the elephants, how they're maintaining. See, I told you there are two kinds of selection process. We are hypothesizing. Okay, we are putting this as a new project actually. There are two types of selection process in elephants: inclusiveness and sexual selection process. Okay, inclusiveness happening in the herd. Okay, the herds are, they are genetically, they are, ants and relatives, they form an inclusiveness. In the case of males, what's happening? I told you the male-female ratio is 1:1. There is no pre-zygotic or zygotic selection process. Every selection process happening after the birth. That's why you got a skewed one ratio of male and female. So males are actually putting into genetic diversity. They are under selection. Get me? They are really under selection process. Uma can contribute more on this.
I just wanted to say that, what did I want to say? I forgot. But basically, the sex ratio is 1:1 when they're born, right?
Yeah.
And when he said inclusive fitness, see, the fact that, just think about it just very logically. Suppose you have some problem with your leg or something like that. Now, if you're a solitary animal, that is going to affect you a lot. But if you are part of a herd, apart from the fact that you share your genes with all of those other individuals, the fact that you're part of a herd, you also might get some help. So the selection on an individual is also slightly relaxed.
Exactly.
And that is something interesting. I don't think it's an idea. It has to be shown, but it's very interesting.
We are doing that actually. We are having a project, we are doing that. So we propose it's an inclusive fitness happening in herds and sexual selection in the males. Interestingly, 1 year back, there was a paper from South Africa, in Kruger National Park. Did you read that paper? They say average age of a bull to breed is 50 years. 50 years. They breed at the age of 50 years. The lifespan is 60, 70. So what does that mean? In young ages, they are not breeding at all. They are a huge competition. You go to the wild, you can find they are fighting and fighting and fighting. Then you can find a huge bull, dominant bull in the herd, and the young ones, when a calf born around 8, they thrown out from the male, thrown out from the herd. Then they will become small boys schools group, then become adult. Then first they have the musk. They try to come into the herd. Okay, they push them away. Okay, because there are huge dominant bulls there. It says 50 years. In African elephants. Imagine. So what is the mechanism behind that? So that's why we said, so what we did, what happened actually, we have a massive poaching the last century. Okay. Like in peacock, I told, the honest symbol of sexual selection is tusk, size of the tusk. So we poached huge tuskers, which are really fit. Okay. Reducing the genetic diversity by removing, this is also a hypothesis we are putting forward. We want to test that in different cases. We are collecting evidence slowly. Yeah.
Can I just interrupt? So he talked about this handicap, right? So the idea is that individuals that have these handicaps, like say these tuskers have these large tusks. So clearly it's not like, suppose you have to run or whatever, it's not a great thing. It will probably slow you down. So potentially that's a signal of something else, because the tusks themselves are not very useful for the individual, right? I mean, having a longer tusk is not much better than having a slightly less long tusk. So the idea is that maybe there are signals of genetic quality, right? That these males who have these long tusks have some very good genetic variation or something like that. So that's the handicapped hypothesis. And what he's saying is, if the handicap hypothesis is true, and combined with human impacts, where we selectively killed off the larger tuskers. Exactly. So what did we do? We removed the best genetic variation from the gene pool. Yeah. So Arun has these, he said, he started off by saying he's a butcher, like totally trivialized himself. But he has these really interesting ideas. And it's nice to see, the earlier session also, we talked a lot about ideas. So it's really nice to see how these ideas, coming from a practitioner perspective, now he's thinking of these really interesting ideas of how this disease is continuing in the population and what will be its impact, right?
Exactly.
Yeah. Sorry, I'm paraphrasing.
No. You're really supporting. So, for example, look at the Chola cavalry in the back 10th century. They had 10,000 elephants, 10,000 bulls. Okay, they wiped out the Eastern Ghat. Okay, so imagine how much genetic depletion happened if the handicap hypothesis is true. Okay, so got a major impact. That's maybe the reason for the emergence of herpes virus. So I'm focusing on the host. Okay, I talk about the pathogen mutation first and environment second. And this is a classical example of the host, we think. This is all hypothesis, but we are testing. We have some projects. We have collaboration with UMA and all. So we are testing that. But in the initial phases, when we did 10x sequencing of some samples, we find that heterozygosity ANDS level is very low in the herpes cluster than the other clusters, age groups. Okay, young age groups. Okay, yeah.
Sorry, just for clarification, do you mean that because they're breeding so low, the social selection is sort of increasing their diversity?
Yes, I will come to that. Yeah, sorry, I missed it because, this is, for example, when you go to, how many hours? I got some more time? Yeah. Okay. So when you go to the forest, if you can find, for example, if you go to like Kabani banks, there are, have you been there? Kabani banks? Okay, you can find hundreds of elephants together. Okay, so during summer. So when a herd goes in Karnataka, that area, especially in Nagarhole, they don't have the practice of burning the carcass. So they leave the carcass and there are bones. Okay, if a herd comes, suddenly you can find a massive mesmerizing situation. They all become silent. Okay, so they touch and using the trunk, they got a vomeronasal organs in elephants, huge, this much size organs. There you can find 2 holes. Elephants are actually sensing the taste of the smell. Okay, right, here I'm coming up to what we are hypothesizing actually. So when a bulls come into a herd, what the female first do? She hold the penis and she touches. If they are close relatives, they would simply abandon. They will not let the animal to breed. Okay, here comes the MHC point. Okay, MHC genes are, they are very specific. Your individual ID. It's okay, unless and until you are, homozygotic twin, it should be different. Okay, so when the MHC actually proteins they're producing, they're undergoing metabolism, maybe in the kidney, we're testing that actually. So these maybe act as pheromones. Okay, there are 2 types of pheromones. Okay, these pheromones may be the reason they are, because I had a classic example for this. Actually, I'm deviating from disease, right? Okay, I had a classic example. In 2019, am I right, Ansil, in Periyar, there was a massive mortality of a bull. A bull killed around 25 females. That's very rare. Bull-to-bull interaction and killing, we get every year, we get 20, 25 cases in postmortem. Females. That's the only classical example. In a season, when we did the postmortem, most of them are pregnant. Okay, so bulls won't do that. Bulls won't kill females at all. So when they come, even though the female is not in heat, when you approach, she sits and will not allow him to mate. Okay, usually that won't happen. This particular head, he just came and punched through the eyes of all the cases. Okay, this is a hormonal imbalance actually would be suggested for that. What I'm saying, there is a sexual selection process happening in females. Females are selecting the males because she is investing the energy, okay, in a lifetime. She's having the pregnancy for 20 to 22 months. Okay, maintaining the baby up to 8 years. Okay, she is investing in it. So she sees that her progeny should be the best one. So that's why the selection process, sexual selection process is important. And that's maybe the reason of that, maintaining the genetic diversity. We have to test that, but could be a reason for evolving emerging diseases in wildlife. Okay, so this is our truly emerging or COIVA, which is we are answering that. Then the problem with the dilemma in wildlife disease ecology is the conservation versus human health. Okay, now we're talking about Ebola, everything, Nipah, and everything. But there is which one is important? Conservation. We're talking about, I think more about conservation point of view rather than other people working on Nipah virus like ma'am doing most of things. So looking at the human health. So this is a case actually. This is called tuberculosis, Mycobacterium tuberculosis. Okay, it's extensively reported in zoo conditions. Okay, you know MTB is evolved along in humans. It's a human pathogen. Okay, there are other MTB, and bovis is there, that is in the cattle. Again, it's evolved from the Mycobacterium tuberculosis from human as a part of domestication. The new species evolved. So MTB is basically a human disease. Okay, so if I'm having a TB, lung TB, I have to sneeze, you know, they form 10 micron particle size, and you inhale, a successful person inhales, and they get infection, but it's getting into zoo animals. Okay, but we got in the wild. The first report of MTB in the wild, Mycobacterium tuberculosis. Okay, so we have massive lesions like this. Now we have 8 cases right now. Okay, so maybe a tip of an iceberg. We don't know what's really happening. So massive lung lesions you can find, the typical TB lesions. Then we did the sequencing, we found it is MTB, and a strain of East Asian African strain was identified. Okay, how this is evolving in wild animals. It's a reverse zoonosis, actually. Okay, this is a kind of interaction, interface in between the wildlife and human. Okay, so can you imagine as somebody can hold the trunk of a wild elephant and sneeze into it? No. Okay, so when we do this training, we find it's called East African Asian strain, which is the original strain actually from the first human evolution, you know, first human migration. Okay, so that's a connection. So that is a reverse zoonosis, I said. Okay, so then emergence of viral diseases, classical swine fever in wild boars. We had a massive outbreak in Parambiculum, and we find that the classical swine fever virus actually evolved in domestication process, not in wild. Okay, so it jumped into wild animals. Now it's endemic in so many populations and destroying it. These are the lesions, classical lesions. And coming to canine distemper virus emergence. Okay, you heard about the stories of CD outbreak in lions, Serengeti lions, and also in seals and yeah, all the species, wild dogs and everything. But we report a massive outbreak of, spillover in leopards. Okay, canine distemper is a human, animal domestication pathogen, mainly seen in dogs. Okay, not in wild. Okay, so. You have to differentiate that when you look into a pathogen, actually. So quite interestingly, I would say, foot and mouth disease, I told you. Foot and mouth disease is actually a part evolved along with the domestication process in the cattle. Okay, it never jumps into wild. We used to have that FMD outbreak in 2000, so 1998 onwards in Gore populations. Okay, suddenly it stopped. You know, foot and mouth affecting the most important trait of a wild animal. What? Walking. Okay. We have a good tiger population, you know. They picked the first one, which cannot walk. Okay. Clearly stopping the virus spread. Like what happened in US, Yellowstone National Park. The brucellosis and the wolf. Okay. Brucellosis was affecting, they got forming the hygroma in bisons. When they introduced the wolf, brucellosis is gone. You know the story of that, right? Rewilding in you don't know? Okay, so Yellowstone, they have the largest bison population, American bison population. So they have the wolves also. Okay, so they have the farms in, now you're seeing the Yellowstone series, okay, in a net. So So what they did, when they have the farms and everything, wolves may be capturing the babies, calves and everything. So what they decided, they want to eliminate the wolves. They eliminate the wolves. Okay. So what happened? Bison population increased. Okay. And there is no grassland, nothing, no green. Okay. So then they have this problem with the brucellosis. Brucellosis is a pathogen, bacterial pathogen causing abortion in cattle, infected to humans also, and most of the vets are positive for brucellosis. So what happened? They cause hygroma in ungulates. They can walk. Okay. So they have a transmission process. It started from domestic animal to wild. Now they started spilling off from wild to domestic animals. It became a huge problem for them. Most of the cattle were infected with brucellosis. They thought introduce wolf. It's called rewilding. Okay, wolf first did, they killed all the animals with the brucellosis. They controlled the brucellosis. Okay, you know what happened in Yellowstone? So many streams started, the forest got grown up. It developed. It's called the whole rewilding concept, actually. We are seeing in the tigers also in our landscape, many streams are, now the population increase, many streams have started, forest is getting back by controlling the herbivore population.
Did you guys get that? Basically, the carnivores exert top-down control of ecosystems, right? So basically, when the carnivores are gone, we discussed one example, the rodents might increase when the mega herbivores are gone. So just like that, the herbivores kind of stomp out everything and they cause the ecosystem to change. But when the carnivores are back, they control herbivore populations to the extent it even affects the habitat itself. So that's really interesting, right? And there's a beautiful example that it actually controls the disease as well.
Exactly. So we have witnessed foot and mouth because the tiger population came up. There is no outbreak of foot and mouth disease. I got just random cases every year, maybe at the border. Okay, maybe because they bring cattle from, we Keralites eat beef a lot, so we bring cattle from Karnataka infected, and most of them are coming through the forest. So some spillover is happening, but not in outbreak proportions at all. Okay, this is a classical example of this thing. It's quite interesting actually when you look into this perspective. Okay, I will tell a small story. We had a huge conflict with the tiger in my landscape. Yes, ma'am. Yeah.
What do you think is the driver for that? Is it through the dogs? Is it through the?
I will come into that.
Okay.
Okay. Because primarily it's seen in leopards. Okay. Because leopards hunt dogs extensively. Okay. Okay. But we got parvo virus in tigers also. Okay. Shetty's work, basically we did together. Dharmavir Shetty and me together. So we find the same canine parvovirus, which is also evolved in dogs, not in wildlife. Okay, there is a spillover is happening. But these strains actually we find the correct dog, serotype. Okay, genotype and serotype. Okay, those correct serotype we found. So leopards, maybe the interface is dog catching. So most of the leopards are living in fringes. Okay, because, why?
Exactly. Yeah, like what has gone up? We just talked about it right now, no? What's gone up? The density of which carnivore has increased in India? Tigers.
Yeah, exactly. You said it. So because I had seen explosion of tiger population in my landscape. When I joined in 2000, I used to get only one postmortem of tiger in a year. Okay. This year how many tigers we captured? 8 tigers coming out. 13 postmortems. Okay. So the population exploded. Naturally exploded. So it's happened in a couple of years back, actually, not a couple of years back, 4 or 5 years back. Our tourist goes into the forest and they take photos, okay? Most of them, they share the photos with us. And there was an increase in leopard population photographs. Okay, then my warden says, my manager says, oh, leopard population increased, that's why. No, they changed one thing, spatial. Because there is a top predator at the bottom, which cannot climb a tree. So they spatially change. In the tree, it's easy to spot a leopard and you can take a postmortem. Oh, sorry, photograph. Sorry. sorry, photograph. So they have a more incidence of this. So we have all the pictures and we analyze. We say this is spatially changing. Leopard is behaving strangely. Most of them are in the estates. Now, in the cities, in Bengaluru city itself, right? So, so they are adapting. Okay, I captured more than 100 leopards, okay, by darting, and most of them I did it in cities, okay, Calicut. You know, there was an argument. Am I diverting myself? No, that's fine. Yeah, okay. Yeah. So, in cities, hard in the cities. So they come out, and because they are very adaptable, leopards are very adaptable species. You know, so there are so many, they say they come from a truck or timber wagon, it's coming. No, you can find leopards in most of the landscape. Okay, they move, there is a riparian ecology, you know that? Okay, in the riversides there is an ecology, it's called riparian ecology. There are small species. So they move, they eat one place, then start moving until they're reaching sea. Okay, most of the cities are near to the seas and the rivers ending. Okay, simple. So this is the situation, and we got canine distemper in wild dogs also. It is quite surprising. You know, wild dog population is really fluctuating. Okay, so Uma having a work on this, right? You did. Your work is basically on that. Sorry. So she's the right person to say it. It's 4 to 5 years you can find the fluctuation of wild dog population. And this could be one of the reasons, but canine distemper is massively affecting. We got so many cases of canine distemper emerging in this beautiful species. And last 3 years, we have massive mortality of civets. Okay, civets and jungle cat. The civet population is really wiping out. Okay, we have the Malabar civet, you know, which was extinct. Okay, when we did that, we got it like, interestingly, we are getting lots of roadkills of civets. Okay, it was proportionally very high. So when we did a postmortem, we find it's a canine distemper. Massive outbreak is going on. When you can do the YouTube, you can find one civet is walking through a street of Kozhikode. Okay, that was, I got the same specimen later. It was canine distemper. It's affecting your brain. Okay, yeah.
That was a picture which initially people said, oh, you know, because of lockdowns, all the wildlife are coming into the city and all.
Yes.
If you see that more carefully, that civet is kind of, you know, wobbling.
Across. Yeah, wobbling across because it's affecting, it's a CNS disease primarily. Okay, so, and when we did the isolate of this and we sequenced that, interestingly, we found that it is very close to hyena genotype in African. It's not with the common dog genotype. Okay, it's very close to African hyena genotype. Why, we don't know. Yeah.
So could it be spelling over between wildlife species, or we may not have them? Yeah.
We don't have the proof, but see, look at the model. See, in lion, African lions, I can expect CD because they are pack animals. Okay, so pack animals are easy to transmission of the disease is much more. Wild dogs, transmission of disease is much more. Look into the, most of them fit into pack animals. Okay, civets are not pack animals, they're solitary animals. Why they're getting? Massive like outbreaks. I mean, last year I did more than 282 postmortems, 282 with canine distemper. Okay, so this actually, so this is something surprising for us, but it's really causing the extinction of the species. And also we got CD from jungle cat also, massive case of jungle cat. So something is, yeah.
So might it not be through mating or like how the societies are basically, if they're monogamous, polygamous, and that is how it's like jumping from individuals to individuals?
Sexual transmission in CD is less because in semen, okay,
yeah,
it's no transmission through the semen being recorded in domestic animals, models. Okay, but there is another possibility. You know why? They are marking their territory using, but urine can have and feces can have. Okay, because it's a hemorrhagic disease, the intestine, feces does have, because we did the feces, we got a lot of cases positive. Okay, so they're marking the territory with the feces and urine. You know, they smell it. And through, it's one of the possibility, through inhalation, they're getting it. There's a possibility, we don't know, we don't have any proof, but we documented in the fecal samples, a lot of cases positive. Okay, am I clear? Yeah. Okay, so these are the, okay, coming into that, then, as a young people, if you want to study wildlife disease ecology, these are the challenges we are facing. Okay, you should know this actually. One thing is that various species with limited knowledge of biology and ecology. Okay, you know the physiology of a tiger or physiology of a leopard, or we don't know the genome even, so we don't know anything about it. Okay, so totally you are ignorant with the biology and ecology of many species. Okay, so when you explore, then only you know. And the second one is ecological interaction between different species, intra-species competitions. We don't know. That's again a dynamics, okay, which can increase host fitness and all kind of emergence. So we don't know about it. The predator interactions, how it's affecting the disease dynamics. I told you about Serengeti, Yellowstone story and our story, you know. So interspecies interactions actually controls the diseases. You don't know much of it. And the classical epidemiological concept cannot be used. Most of the epidemiological concept, for example, SAR model, is actually for humans. You cannot put that thing in wild, okay, in emerging diseases. Very, very, very difficult, okay, because you don't know how our reservoirs, how are susceptible, how much are infected, okay, you don't know. You don't have any data. So SAR model, the classical epidemiological picture to put in is very, very difficult in wildlife. Okay, then fourth, conventional surveillance methods cannot be used. Okay, so for example, we use, okay, for example, in the case of, we can do the serological, survey methods. Basically, we look into antibodies, right? For you to see the population, which is, for example, Dr. Guha talked about coronavirus. You know that the antibody, you know, naive population is decreased, and slowly you have the antibody level, so the disease comes down. By looking into antibody level, you can see the population is how much is susceptible and susceptible. Okay, so that's why we are getting spikes of the disease. Okay, the corona, we are expecting the same. There was a spike of antibody, so the disease goes down. Then the naive population comes down without antibody, then you can expect a spike again. Okay, so that's why it's become endemic. And slowly they co-evolved. Okay, so imagine every pathogen's feeling is that it should survive, it should propagate. It kills its host, pathogen is gone. Okay, so slowly they become accustomed and they become, what do you call, co-evolved. That's why I said that niche concept in the beginning. The pathogen becomes slowly co-evolved and become endemic to the population. And finally, it's a controlling factor. Then coming the evolution, and it is controlling the population density and genetic diversity. Okay, that's the concept basically. So conventional surveillance methods cannot be used in for example, the passive surveillance method, because you cannot examine the animal. Can you examine a tiger in your population? No. Okay, only one chance for me is butchering, post-mortem. Okay, so bias sample, because most of them are putrefied. You won't get pathogen. Okay, so that much you have to involve. So you cannot do a zero surveillance in case of wild animals, which is the toughest portion. You can do the cross-sectional study like Ansil did in bats. Okay, you can do a cross-sectional study. Okay, aggressive cross-sectional study, not a passive study. Okay, so surveillance methods cannot be used or whatever in the classical epidemiological terms. Then conventional laboratory tools. Okay, we are looking into most of the cases for the surveillance. We are looking to antibody ELISA. Okay, can you use a human ELISA into wild animal? No, the antibody is different. You have to develop ELISA for that species itself. That's exactly what's happening with the herpes virus. So now they develop, because they cannot culture the virus, so now they develop a new ELISA kit for herpes virus. Okay, I cannot use simple classical simplex herpes ELISA in elephants. Okay, so only one thing left to you is you cannot even capture the animal. Do you think you get a permit to see what your sample says? 10%. Okay, basically. So I have 10,000 elephants. I need 1,000 elephants. Do you think you get a permit to dart all these animals and get the samples? No. It's a conservation priority and it's a different issue. So these are the challenges you are facing. You cannot use most of the conventional laboratory tools. That's why we are focusing on molecular methods, okay? Because you can do it, it's versatile, okay? So we are having the next generation sequencing facility, metagenome, so that's why we are applying into this area. Okay, so lack of previous research data. Show me a paper, except some of our paper, on wildlife disease ecology. No? How many PhD theses? Maybe mine is there. So a couple of them. So very ma'am's there. Ma'am's PhD is there. So yeah. So on wildlife disease ecology. So there is no data. We are standing in a plethora without any data. So that is a realist problem. We have to develop from the start. Okay. So yeah. Okay. So this is the problem. See, most of the time we're getting carcass like this. So highly putrefied, you cannot get any samples on it. Okay, we get non-invasive methods. We develop a new technique, drilling the bone, getting the marrow, and getting the pathogens. Okay, because the marrow is encapsulated in the bone. So we develop that. It very works with a herpes virus, even 1 week old carcass. And sometimes you're lucky enough to get this kind of carcass. This, she was, the calf was alive, she was dying. Okay, she died in front of me. So even though I tried to save the animal. Okay, the next one is urban wildlife disease ecology. This is something is quite important. Okay, so urban wildlife is, there is a huge ecology of urban wildlife. There was a debate, basically, I would say, in Kerala Forest Department. There's a huge problem with peafowl in Kerala. Okay, the population is increasing like anything. It became a pest in many areas. And there was idea that they say the climate change is happening. Actually, no. Basically, there is no predator for, it's an egg predator. You can just crash the egg and you can control peafowl population, So brood predators are less now because of vanishing of urban wildlife. Okay, so there are indications. So this is something, this is a transmission mechanism. For example, in the case of rabies, imagine it is there somewhere in a niche, maybe in a bat or maybe in some rodents, okay? Something hunted that, then slowly it spread. It came to urban wildlife like jackals, okay? The jackals got it, and to the dogs and to the human, okay? So this interface is also very important and focused because this is in between the mediations, in between the niche wildlife disease ecology into emergence, okay? So very limited work is happening in urban wildlife disease ecology. So, okay, so what are the real, for example, in the case of viral reservoirs, I mean, so why some species can do this? Okay, most of the emergence happening in rodents and from reservoirs are rodents and bats. Why? When you look into bat genome, there are receptors, especially viral receptors. Okay, there are 9, 10 types of viral receptors for each species of virus. Bats have most of them. Okay, in the Paramyxoviridae group, Filoviridae group, bats have the receptors. What does that mean? What does that mean? I told you at the beginning, every pathogen has an idea that they have to maintain. Okay, so bats' population size is huge. Okay, so they can adapt easily with the pathogen. Okay, that's why we have hundreds of pathogens in the bats and the rodents. Okay, because they have the bats has to transmit them in between. So it becomes a whole pool of the virus. Okay, it becomes slowly adapted, and bats are, so that's why there are lots of theories. Okay, why bats having this much of, even fever hypothesis. They say that because of the virus, it creating a high temperature. That's why bats are the highest BMR. You know that, basal metabolic rate. They are maintaining it because of the viral level. Okay, there are so many hypotheses putting forward. Anyway, this kind of evolutionary receptor homology and an evolutionary process is something important for emerging infectious diseases. Okay, that's why we have more emerging rodents and bats are the reservoirs for emergence. And then again, special as a viral adaptation also. This is Nipah. We had a first Nipah outbreak in 2014. Then we had 2018 and 2022. We are actively involved in Nipah surveillance. So in Bangladesh, actually, what they will do, they have this palm toddy. So okay, they drink Okay, the bats drink and they urinate. Okay, so they have the highest viral load in the urine. Okay, and yeah, so this is a unique immune system. They have a high adaptive immune variations, innate response and adaptive responses very high in the bats, so they can sustain. Okay, the antibody level, they balance the viral titers. A lot of papers. And regarding the emergence of Nipah, actually, so I'm working with the Nipah group. So why it is happening, why it was emerging is basically there were so many hypotheses. One among them actually we found, there was huge cashew cultivation was in Kerala. Okay, now it's gone. Okay, so most of the time people prefer to have bathrooms in the house because you can have tons of cashew nuts, you know, in the near to the roofs. Now the cashew is no more in Kerala. Okay, and again we have a huge orchard system now from you know I mean what do you say, exotic fruit plants. Okay, which may be the reason, because the first outbreak we find that it is from mango. Okay, first outbreak in Nipah in Kerala. Then we find that from arecanut. Okay, because they chew the arecanut. Okay, so the interface level is improving because the change in the cropping pattern may be a reason we put forward. We are trying to analyze this data spatially and this also. So this is the end of it actually. So wildlife disease ecology is clearly interdisciplinary. Okay, it's a jack of all, as Dr. told. So so there is a role of veterinarian, there is a role of biologists, there is a role of ecologists, there is a role of genetics, there is a role of, you know, everything. So that's why, I mean, I think we had a discussion. I mean, who had a discussion with the Dr. Guha? So that's why people are confused. Okay, so a programmer can do a lot. Okay, so mathematicians can have the models, and it's a mix-up of science. And the collaboration is really needed. Okay, it's not a one-man science. That's why we have a huge collaboration with her and so many other groups. We have a collaboration in US, UK, and everything. There are mathematical model producing people. Okay, we have bioinformaticians. We require massive need of bioinformaticians. Okay, we have a huge problem to question, answer many of the biological process or ecological process we need to analyze, because most of the bioinformaticians doesn't know what really happening in the field. Okay, we know what's happening in the field. To transcribe that knowledge into sequences and putting into real phenotypes, you know, you need different tier of people. Okay, so this is the crux actually. While it is everybody can contribute into it, everybody has a role in it. Okay, thank you very much for this.
So before the others, then you ended saying it's so interdisciplinary. And so basically where to start then?
Yeah.
Sorry, maybe that's difficult.
I think it's see, I started my career, see, my role actually basically to dart elephants and, you know, triangulate tigers, mitigation of conflict and everything. But when I'm interested in ecology, so this is my transformation. Basically, I didn't study anything in vet school. Okay, they taught us not elephants at all. They taught us like cow, dogs, and everything. So, but it's entirely different when you're doing the wildlife disease ecology. It's not. It's a natural selection process. Okay, your concept is changing because other one is artificial selection is happening in domestic animals. You artificially select. Okay, a breed, a dog, your pug is, you know, it's selected artificially. But here is natural selection is happening. To understand the crux of natural selection, change your perception. Okay, so you remember the word, I mean, without the knowledge of evolution, nothing makes sense, by Theodore Dabansky's words in science, actually. So once you have that evolutionary concept, your whole perception would change and your view would be changed. That really happened to me. Okay, so my transformation as a vet from to here is like basically now a biologist. My PhD is actually in wildlife biology, not in veterinary. Okay, because when you transform yourself, so that's a process. That's in the field. So once you have I don't know, you differ from different disciplines, right? Okay, so when you have that perception onto this ecology, then definitely you will receive kind of perception. I'm thinking. Am I answering?
I was thinking also more because there is clearly the wildlife aspect, there is a immunology aspect, there is a ecology aspect, there's genetics, and how to, like for younger people, like how to, like where to start, and is there a way to, without teams, can you actually make a contribution, or you should focus more on building collaborations and getting a team?
Exactly. That's almost said it. So look into wildlife disease papers. Okay, last 10 years. Okay, you can see a handful of them. You have thousands of papers on zoo animals in captivity. You have, but you don't have a real paper on what's happening in wildlife diseases. Okay, so very. So one thing is that it's a naive science. It's just emerging science. Okay, so I was lucky that in UK, a couple of my professors are really pioneering on that, like Peter Deshak and those kind of people. So actually they gave me, they changed my perception actually. So I was lucky enough to have them to guide me, to change my perception. But look into that, there is no other publications. Okay, so there's a huge field we can work on. Okay, I can simply tell a story about this. The social aspect of this. So I told you we had a huge conflict happening on tigers in Wayanad landscape. So with a massive tiger population in the world, actually. So it started in 2012. Okay. So there was a first case. I'm telling a story. Okay. Do I have time? Yeah, you have time. So in Karnataka, they captured a tiger, problematic tiger. Okay, so we classified tiger conflict into many, 1, 2, 3, 4. One is it's called slaughter group. Young animals, they cannot survive. 70% of tigers are killed by the commodity males because they are territorial. Okay, so young animals they have to find, because it's saturated. Most of them goes to private estates and everything, which is abandoned. Most of them. And there are other one is habituated, which are injured animals. 13, 14 years, the other tiger will attack. They come out. Okay, there are different patterns of it. There are many of the man-eaters, and we have another group which is infected, disease infected, like lung infection, so many other flukes, and so many other. So our conflict starts in 2012. Okay, first the Karnataka government captured one tiger. They released the tiger near to Wayanad Wildlife Sanctuary, and this is our habitat forest department. Okay, no, in the border, and the border Kerala to our landscape is nearby, only 13 kilometers. Okay, to Karnataka, another city is almost 300 kilometers. 120 kilometers is there. So they came to the town, Sultan Batari town, and is starting killing animals and attacking people like that. And I darted and I captured. Okay, my boss said, okay, release back into Karnataka, into the Karnataka. So it's just like a exchange type of thing. So I released back near to the Karnataka. Okay, it came back into human settlement again, and it was rampaging like anything. Then finally I darted him again. Suddenly it attacked one of my staff and it shot the animal. Okay, so it is almost 15 days, 25 days, the whole highway was blocked by the people. Okay, they were protesting, they're blocking. So once I'm coming to Sultan Batari town, to my office, I saw small young kids actually parading and with the boards and everything, kill the tiger, safe from the forest. Okay, and there are teachers behind it, and I feel so shameful because they are the new generations. Okay, they are supposed to protect the wildlife and they have to understand. So we have nature camps. So our nature camps, there are 1 week program. The schools can come, selected kids can come, then we'll have lectures and everything. We take them into the forest. It's basically to build the kids. But these nature camps are mainly focused on rich kids. It's mainly happening in cities. Okay, right? So I told to my managers, so we told we have to change this. We have to have this kind of nature camp to those kids and who are living nearby. So we changed the pattern. The first thing I specifically called this school for the first camp. And I was talking vigorously about conservation and everything. And a small girl, first standard or second standard, she stood up and said, so why we need tigers in the forest? We have tigers in the zoos, right? Why need the tigers in the forest? Simple question. Okay, then I was just puzzled. Then I asked the same question to the teachers who were sitting in the back. And I told, why we need tigers? Then one teacher said, we need food web. Okay, so it's a predator. What food web? Tiger with. We are eating the rice, chawal from the, rice field and fish from maybe from sea or something. What is the tiger's role in the food web of human being? Nothing. Nothing. Is it done? Can you suggest one? No. Okay, then I told, okay, we can do one thing. We can shoot all the tigers and leopards from the forest, okay? And what will happen? Then they said, herbivores will go. Then what will happen? They will destroy the forest. Okay, okay. Then, okay, we don't need the herbivores even. We can kill the herbivores also. We don't need anything. What will happen? What will happen? The forest will collapse. Okay, if you go to Puerto Rico, you can see crushed forest. The first thing is the most, when you see it, most of the time people look into the forest saying, oh, beautiful. Actually, huge competitions are happening in between, just like a tiger chasing a deer. Okay, trees are competing. Vines are, the most dangerous is the vines. They go, they cover the whole top, and forest dies and crush. To control that, we need herbivores. To control that, we need carnivores. Then only we have what? Forest. What for? Water. Okay, so forests are storing the water. Then we have the rivulets and we have the rivers. Not like in North India. It's not glaciers actually supporting. The forest is supporting. Because without the forest, there won't be any water. In my city, where I'm living, in Calicut, I can have air condition and everything. Okay, I can put it on. But I don't have any problem with the wildlife. Who is facing the wildlife? Who is living along with that? Okay, this is simple social aspect of wildlife. So when you come into that, these things really change your perception. Hope I make sense. Yeah, please.
This is not a disease ecology related question, but I'm just very curious because with respect to this tiger population increasing in Wayanad and everything, since you are from the Kerala Forest Department, so I just want to ask you, what do you think is the main reason for this population explosion of tigers happening? Is it actually happening, first of all? Or is just as people say, the estate owners making issues about it?
Okay, so two things. One, Uma is the best person to talk about how tigers came to India and how the population formed. Okay, she got much diversity towards central India, am I right, Uma? And less genetic diversity towards south, as I told previously about the elephants, because that's why you don't have tigers in Sri Lanka. Okay, because the gap formed. Okay, tiger, I would say it's an invasive species. Am I agreeing? Are you agreeing with me? It's really an invasive species. We had lions. Okay, that's why lions were in Singapore. That's why Singapore, the name, Singapura. Okay, the lion's gone, possibly due to tiger and the human invasion. Okay, meta population formation. Interestingly, one of the students came 1 year back. She wanted to study black buck population in Tamil Nadu. There are 5-6 populations are there. Okay, so when we examine that, they are highly genetically diverse. Okay, these 5 populations. One is in Gindi, one is in Nagarhole, no, Satyamangalam. Okay, so like that, she collected the fecal sample, we extract the DNA from the fecal sample, we look into D-loop region of the mitochondria, and we found that they are different extent. But what I'm saying, tiger might have a role. It's adapting. Okay, so there are evidences. In the beginning, we have Pakmak sensors. Okay, now we have camera traps. I know how many tigers are living in my wildlife. Okay, we have a clear data with us. Okay, each animal, each individual. That's why we are saying we found that habitual conflicts animals which are aged. I have data from 2017 onwards with us. Okay, I can identify each tiger in Wayanad. So very clearly it's exploded because there was not, I told you about the postmortems. In the beginning, I used to get only 1. Okay, now the conflict is huge because the population size has increased. At the time, there was a huge herbivore population. Okay, you can see hundreds of goats in a herd. Okay, hundreds of spotted years in a herd. Now, I think literally I would say goats are wiped out in Wayanad. Okay, so when you have, this is a home range concept, you know the home range concept. So when you have the home range, which sustains, for example, 5 square kilometer is enough for a tiger to sustain in those times. As the prey population gone down, so they expanded the population, and there is a chance for fight. So once they are injured, they come out. So we classified the population like that. There is the first group is called floaters. Okay, a tiger cub born around 2 years, it separates from the mother. Okay, the main predator is tiger. Kills, simply kills, and eat also. So he will roam around for a home range, but all of them were occupied by superior animals. So he tries to goes out. So there is another problem within Kerala also. Price gone down for cash agriculture products like rubber and coffee and all. So what happened? Many estates are abandoned. Okay, so they goes and occupy. This is called floater group. Okay, second group is told habitual conflict animals. Clean pattern with them. These floaters become slowly adapted to human settlements, and they started doing the picking. Second group is the injured. Okay, when it's 13, 14 years, they have all kind of injuries. The other one goes and smash him. And with most of the injuries, I just recently captured, 3 days back, I captured one tiger. And with a huge humerus fracture, they will come out to the periphery. Okay, the behavior is quite interesting. They will not capture, we have around 6,000 cattle inside the sack. They will not pick it. They all pick animals which are tied in the stable because they are not competent enough to even to do that. Okay, they just come and pick the animal from the stable. Very simple management to capture them. When you have the carcass, okay, put that in a trap. Within hours, the animal will come and you will get in the trap. Okay, then there are another group is called sneakers, which have diseases just like that. The pattern is entirely different. Most difficult group. So those animals are really there is lots of lung infection is called Paragonimus westermani, like fluke infestation. So which kills the lungs around 70-80%, so they cannot run. So they become this kind of strayer group of. There are so many. Even current December is also one of the reasons for that. And fourth one is the man-eaters. Okay, so we hardly don't have much man-eaters, only 3 cases, and we shot them all dead because you cannot handle. They only pick human. So these are the. So I hope I get your answer. We have a huge data bank with a camera. We know each tiger, just coming out and.
Sir, as ma'am has pointed out that collaborations are important.
Yeah.
So are you in connection with some vaccine industries there we can introduce some intervention to contain the diseases or some issues like I think like this herpes virus we can make vaccine.
Yeah.
So and there are like vaccine manufacturers in India like Hester or IIL who can make vaccine for you and it is we can work on some common like easy to make vaccine platform like subunit vaccines, which can be used for many diseases. Like just we have to change the antigen sites and we can adapt that for quicker response.
How we can apply that in wild? That's a question. Okay, so there are bait vaccines. For example, rabies.
Yeah.
Okay, in US they develop a oral vaccine, bait vaccine. So you can put along with the meat and you can throw it and. In those conditions, how you can administer those things in the wild, and how can you monitor and make sure that you got the proper dose of it. You get me? I can do a bait vaccine. How can I make sure that the same?
We can try something like bait, like you said, rabies thing. We can figure out the way we can give vaccine or vaccinate the animals.
That's a difficulty actually. Now only, last week only the new vaccine came for EHV. Okay, last 15 years there was no vaccine. Last week they tested the vaccine in Netherlands. Okay, in Amsterdam, no, other one? No, Amsterdam, no. Yeah, Amsterdam Zoo, I think, yeah. They tried the first vaccine. Okay, so because they cannot cultivate this virus, no cell lines available for that. So without massive protection of virus, how can you create a vaccine? Okay, that was the challenge they were facing, but they overcome that, especially due to subunit vaccines they developed now one. So, but the problem, the most of the vaccine cannot be used in as bait vaccines.
Yes.
That is a problem. So that is a technical issue. So the ethical thing is that how you see this as a problem. For example, CD in civets, I told you, it's much more related towards hyena strains. How you connect that? So how do you decide which mechanisms are actually driving the emergence? That is a primary challenge we have. So I told you the host, environment, and pathogen. Okay, is it due to host genomic problems or host immune issue, or due to a pathogen mutation? Okay, or due to environment change, as I talked about Clostridium. So how it's actually driving, which is a real driver for emergence, and which is the first question to be answered. Okay.
So I have a very basic question. So you are discussing about breeding of elephants is only 60%.
Females. Females are breeding.
Yeah, females. So why can't we do artificial insemination in wild animals?
I'm talking in the wild. I'm talking in the wild.
It's not. Is it not possible? Like
no, there is only one insemination in elephants in the history. That is the mother of Kumari. I told you the first herpes case in 1995 in National Zoo in Smithsonian. So that's only case. It's because it's very difficult in elephants. And anatomically also, because its vagina is like this. So what they did, they drill a hole, okay, and through that they inseminated. So you cannot have a flexible thing. And the hormonal pattern is different. LH surge is different in elephants. It's literally impossible to breed, artificially breed, in wild especially. How do you monitor the cycles and everything? It requires almost 2 years to have a plethora of getting the cycling situation, then you can do it. So you can introduce the bulls, genetic variable bulls, to increase the heterozygosity. You can try that.
Okay, thank you so much.
Thank you very much.